Hello, in this video I'm gonna show a demonstration of meiosis, okay? So you can try to replicate at home. Here I'm gonna show just the chromosomes and the cell membrane or nuclear membrane, okay? I'm not gonna show the organelles or the enzymes and everything else, okay? I'm gonna focus on the chromosomes only, but be aware that's not these that duplicates as well. Okay, if you want a more deep explanation, I have another video when I show mitosis and the interphase more detailed. Okay, so here I have a cell. It has just 2 chromosomes, okay, or 2 pairs of chromosomes. So I have in total 4 chromosomes, but 2 pairs. The blue and green I represent the chromosomes I received from my daddy, and the red and pink from mom. Okay. Okay, so to start a duplicate, cell division or meiosis or mitosis, I always need to go to interphase. Okay, so I'm not gonna detail the interphase here, but what we need to focus, what the most important thing that happens in interphase, the duplication of my DNA. So here I have my DNA, okay, in chromosomes, but I'm gonna duplicate it. So I'm gonna make a copy of each of them. So here, in interphase, I make a copy. Just remember, it's phase S of interphase. Another copy for each of them. Okay, so that's what we do in interphase, just a copy of them. So here now I still have 2 pairs of chromosomes, but they are duplicated. Okay, that's it for interphase. And remember, the chromosomes are not condensate yet. They are just like disorganized and not condensate yet inside the cell. That's the interphase. After the interphase, I'm gonna go over the first process, meiosis 1. And what's the first phase of meiosis 1? Is the same as mitosis, is prophase 1. What happens in prophase in mitosis? Condensation, right? So, for example, here I'm gonna organize, they are gonna condensate more organized. So I'm gonna organize my homologous chromosomes together. So the small one, the big one, they are now organized. And the other thing that happens in prophase 1, the nuclear membrane disappears. So I'm gonna erase my nuclear membrane. Okay, so that's it for prophase. After prophase, what do I have? Metaphase. What happens in metaphase? Middle. The difference here between meiosis 1 and mitosis is because in my metaphase, I'm not gonna do just a single line of chromosomes as in mitosis. I'm gonna do a double line like that. So my homologous chromosomes that are duplicated goes to the middle. And remember, when I duplicate, I call each copy of this as sister chromatid. So here one sister chromatid, here another one. So I have these chromosomes duplicate, so in this chromosome I have 2 chromatids. So in metaphase, it goes to the middle, just like that. And goes to the middle as tetrads. Okay? Now, what's the next step after metaphase? I have Anaphase. Ana stands for separation. So, separation for what? I'm gonna separate my homologous chromosomes. So here, I'm gonna separate the homologous chromosomes. So they goes for each side of the cell. So that's it for anaphase. After anaphase, I have telophase. What happens in telophase? My nuclear membrane appear again. So my chromosome decondensates, so it's gonna be disorganized again. And I'm gonna do another nuclear membrane. So disorganization, decondensation, and another nuclear membrane appear. And now it starts the cleavage between these two cells. Doesn't cleavate yet, but starts to cleavate. Okay? After telophase, how do I finish? With the first cytokinesis. What happens in the cytokinesis? The cell just splits in two. So I'm gonna redo this. I'm gonna redo this. And this one. Okay, so in cytokinesis, I complete my split of the cells. And that's it for meiosis 1. So in meiosis 1, I create two new cells, two daughter cells, with half of the chromosomes, okay? Instead of have one pair, I have just one for each duplicated. That's meiosis 1. Now I go over meiosis 2. What happens in meiosis 2? What's the first phase? Also, prophase. What happens in prophase? The usual. So, my nuclear membrane disappear. And my cells, my DNA condensate. So, I can see the chromosomes again. So, that was disorganized. My nuclear membrane disappeared. And my chromosomes condensate with the help of histone, remember? The protein. Okay, so that's the most important part for prophase. After prophase, what do I have? Metaphase. What happens in metaphase? Goes to the middle, the same. But now I don't have the two homologous pair. I just have one each. So I'm gonna do one single line like that. So one single line. Here you can see my daddy chromosomes went to the one side and my mom goes to the other side. So I split the homologous chromosomes. Now they go to the middle again. After metaphase, what do I have? Anaphase. What happens in anaphase? Separate. Now what I'm going to separate? My sister chromatids. So my sister chromatids, my copy, goes each for one side of the cell. Here again. So I separate my sister chromatids. After anaphase, again, telophase 2 now. What happens in telophase 2? I form again my nuclear membrane. So here I have two single chromosomes. So I'm gonna form two new nuclear membrane. Another one. And I start my cleavage. So here, I didn't finish, I just start the cleavage. Here, and this one. Okay, so that's it for telophase. And finally, I finish with the cytokinesis. What happens in cytokinesis? The complete cleavage. So my cell will completely separate in two. Here I have one. Another one. So here I formed already 2 cells. I'm gonna cleavage this one as well. And that's it. So how many cells do you see now? 1, 2, 3, 4 cells. And look at the chromosomes. This one looks different from this one as well. For example, if I have 20, in this case I just play with 2 chromosomes, but I have in humans 23 pairs. So all of these new cells that I formed are going to receive a new combination. So this is increase the genetic variability. Because for example, let's say this one forms a baby and this one forms another baby. They have the same chromosomes. No, they don't receive the same. That's why sisters and brothers are not identical, because they receive from their daddy different combinations of chromosomes, okay? Okay, so that's it for meiosis. I form 4 cells, not just 2 as in mitosis, and I form them as half of the chromosomes that I have before. Not the, for example, if I have 23 pairs, I'm gonna have just 23 single. So in this case, I had 2 pairs or 4 chromosomes, and I had each cells with just 2 single chromosomes. That keeps me, helps me to keep the same number of chromosomes in the species. Okay, that's it for today. I hope it helps, okay, for you to understand better this process of meiosis. Thank you so much.